Right, so it's 10th of December 2024. Open Source Antibiotics Mur Ligase meeting. And thanks for putting the agenda up online. Yuhang, that's great. That is I mean, obviously the stuff on the agenda is a bunch of stuff that we've got to keep an eye on. But I think that there are key things today for us to talk about. And I think Yuhang's update is pretty short because he's been wrestling with HPLC for a couple of weeks, doing some sterling work on getting the machines back up and running. And they are now up and running, and so he's able to purify another few compounds for shipment to Warwick in the coming, well, hopefully before Christmas, I guess. Yuhang, is that the hope?
Yes. Another two to three, if I manage to get them right. Yeah.
All right. And essentially, these are the compounds that we need to complete the grid for the paper. That's ultimately.
perfect.
Yeah.
So microbiology and the full spectrum of ligases, or just MSC?
Yeah. So I think when you ship the compounds, just make sure you also ship that deck that has all of the kind of data that we've got, which you've been summarizing. Yeah. And then we can be sure that we get all the data that are going to be needed. So there are no holes that referees will pick holes in for the paper.
Yeah, that's what I've been concerned about. Yeah.
Yeah. Okay. All right. So that's incoming, but it's been an equipment problem we've had.
Yeah. Because
Sorry. You did some data. You did some experiments on the solubility of the compounds. Is that right?
Yeah, we did. Did you want to talk about that?
Was that done with the compound in different solvents, or was it only done in DMSO or something like that?
I think so we had some compounds measured, but those were in one of Guy's series.
Yeah.
Okay.
Not the AZ series.
Mm-hmm.
I think, Yuhang, we've never had any issues with solubility for the AZ compounds, right? They've always been
they're very polar and water soluble, so shouldn't be very much an issue.
When you say water soluble, how much can I go in the concentration if I try to solubilize it in water?
Warm water.
We don't know a number.
Yeah, we don't know exactly the number, but we don't know. But every time when I purify them, they're from reverse phase column, and
so within the context of assays, all the compounds that you've sent to me, the pyrazolapyrimidines, they all are perfectly happy up to 1 millimolar at least.
Right.
I've not had an issue with them like I've had with other compounds. So they've been reasonably well behaved. I mean, you have to bear in mind that they're made up in stocks in DMSO, and they're added in DMSO, and there'll be about 0.5% of DMSO in the actual assay, but that's probably neither here nor there. But up to 1 millimolar, I think essentially you're safe.
Okay.
That's fine.
Well, I'm just asking because I don't think DMSO is doing anything good. For a different project involving Mur Ligases, I've got some structures, and that didn't have any DMSO. So I think we have to remove it completely. And I was just thinking of starting with water instead of anything else. But if you had any data, obviously, then I will follow up from that. I've got some data set with one of the W compounds, but the resolution is not good enough, and I don't see any blobs that resemble any compound. Yeah.
Okay.
Just okay. I'll be careful, and maybe later on I only ship the solids instead of in DMSL stock.
Yeah, that would be good.
Yeah.
Okay. Thank you. Yeah.
Yeah, so I mean, about this, we did have a residual worry about some of the compounds in the other series. So, some of the compounds that we looked at previously and some of the compounds in Guy's series. Guy, did you happen to have the solubility data to hand just so that people had seen that? Because we had it measured twice, right? We had it measured in Barcelona and by a company.
Yes, not all of the compounds actually. I actually put the data on the GitHub
Oh, you did.
page
Sorry.
already.
Okay.
Yeah, let
Right.
me. I can just a quick open here and show. Yeah, I mean, I didn't send all the compounds, all the first series that I sent initially to Warwick for testing, because I didn't have enough material for all of them. So I just sent like I think maybe like 6 or 7. Yes, so yeah, I can share it quickly if you want.
Yeah, great. Yeah. We were concerned that maybe some of the lack of activity of a couple of these compounds were because they weren't soluble enough, but it looks like the compounds are soluble enough. Not as bad as we thought.
Yeah.
Can you see? Yeah.
Yes.
Yeah. So
yeah. I only sent 5 basically. That's the only ones that I had enough material to spare some.
And these values were kind of backed up by by Analyzer, who analyzed a couple of them and came back with similar results, right?
Yeah. Yeah. Especially 60, 70, and 75 and 78. Yeah. They're. I mean, it wasn't exactly the name numbers, but it was pretty close related.
Yeah.
So it was quite reliable. Yeah. So yeah.
So yeah, in this case, the lack of activity is something to do with the compounds rather than
solubility. Yeah.
Yeah.
Right. Yeah.
Right.
I'll stop sharing.
But yeah, that's useful to know though. If sending them as solids will be better, then that's great to know. Yeah.
Yeah, I think so. Yeah.
Yeah.
All right.
Yeah.
Okay. Thanks. So so with that said, Warwick team, did you want to present anything that's been happening in the last month?
Yeah.
Yes, Henry.
Yeah, guess that brings it to me. Although I've been here for a bit more than a month, so that's all.
Right.
I'll basically just be going over what I've been doing for the past few months as part of my master's project, and for some other involvement before that as well. Okay, I'll start sharing. That should be fine. Yeah, so
Can they see that?
just to check, can you see this slideshow? Yeah. Yeah, so this has been a project I've been involved with for my master's project. Was aimed at developing multi-target. It's aimed at developing compounds which have multiple targets both within the different Mur ligases and at different binding sites on the same Mur ligase enzymes. So the starting point of this is generally the observation that the 4 of the Mur ligases, Mur C through Mur F, have quite significant similarity in their substrates, in their domains, and their mechanisms, and in their structure. So it's being described which domains interact with the different substrates, and with some previous work has been demonstrated that, for example, in ATP analog such as ADPCP has been able to effectively inhibit all 4 of these Mur ligases from Pseudomonas aeruginosa. So the idea kind of evolved from that towards if there could be this shared inhibition at the ATP binding site, could there also be compound which targets that and potentially, say, the UDP binding site or the amino acid binding site? Now, the starting points for this was looking at this series of dinucleotide polyphosphate compounds. They consist of 2 nucleotides joined by a phosphate chain of variable lengths. They are found in basically all cells as a byproduct of amino cell tRNA synthesis, especially AP4A. But importantly for us, really, they have a structure that resembles broadly the two of the major substrates of these Mur ligases. You've got ATP at one end and AUDP Murnac phosphate, Murnac peptide on the other end. And this is especially true, as was initially demonstrated with UP4A, which has a uridine at one end, which might be expected to mimic the UP Murnac, and an adenosine at the other end. Now, this was our initial data set with just using UP4A as a compound, and we identified that against Pseudomonas, Mur was by far the best target for that as a kind of pilot result. So moving on from this, we intended to look at the other. We intended to look at other dinucleotide polyphosphates. One of the main advantages of this is that we've been able to get syntheses for these done with quite variable phosphate chain lengths. So the hope is that if we are able to vary the chain length, maybe if these native Mur ligases have these different sized substrates, we might get a relationship between how effective these inhibitors are and which and against the different Mur ligases. So using the Amplex Red assay, we managed to get IC50 values for a bunch of these. The areas where the bars are missing are where they were too high to be really reliably measured. And the general trend for the compounds between 1 and 6 phosphate chain in length in the chain has been that the longer the chain is, the better the inhibition against really all 4 of the Pseudomonas Mur ligases. This is it. And between the Mur ligases, again, Mur was by far the best target for this, especially going down to some quite low, some nanomolar inhibition for the GPs for the 6 and 7 phosphate chains. And Mur-D was also showing some signs of being a good target for the longer chain polyphosphate that we've been testing, or have to do more kinetic experiments on that front. And with this encouraging result of this powerful inhibition, we wanted to figure out the inhibitory mode for this. And broadly speaking, it was very clear that the inhibitory compounds that we had were competitive against ATP, and they, perhaps with slightly less confidence, but still being quite clear that it seems to be non-competitive against the UDP substrate for Pseudomonas Mur-E. Again, with this model, we are able to get Ki values for these top 3 phosphate chains that we've got. We've ordered some UP8A, so with 8 phosphate chains, but it hasn't arrived yet. Once we get that, we're hoping that it would also be a potent inhibitor on this front. And from this, we've also looked at whether this inhibition is conserved across different bacterial species. So primarily these, not going from Pseudomonas, we've looked at 3 other Murs, so from tuberculosis, from E. coli, and from strep pneumoniae, which is a gram-positive Mur with with some differences in substrate. And general pattern is that Pseudomonas appears to be the best target for these dinucleotide polyphosphates. And strep pneumoniae, interestingly enough, is a very bad target, in fact. So this is kind of the extent of what we got with the kinetic data. But to get a better idea of why this is going on, we looked at some bioinformatic approaches using some docking programs that have been developed recently, especially so from these sources. And primarily, we've used this program called Try Discovery, which is which was kind of released a few months ago, in fact. So initially, we did some experiments to see how well it matched up with known structures. So this is a crystal structure of Mycobacterium tuberculosis with two of its native substrates, and in blue and in green is the predicted structure using our program, using the program. And while there are some differences perhaps in the amino acid binding sites, we deemed that it was sufficiently similar to at least use to generate some predictions. Okay. And indeed, when we used it to try and replicate our experimental results, so this is with Pseudomonas Mur, we got results that were broadly consistent with our kinetic data. So the dinucleotide polyphosphates were predicted to bind in the ATP binding sites, being competitive against ATP, while not really reaching the UDP sites, which reflects what we got from our, which reflects a non-competitive mode of inhibition. And we were to run this through PosEdit, a program to predict these, to generate these likely interactions with the residues on the proteins. And the general, to try and get an idea of what interactions are key to this mode of inhibition. And what we observed was that at the ATP binding sites, it was fairly universally conserved, both across different species and across different Mur Ligases. They all had very similar number of interactions. Really, what was different, however, was the interactions at the UDP binding site. So, as an example, this is a comparison between Pseudomonas and E. coli maize, both with the same EP6A polyphosphates, and we've got this movie where in the E. coli it's been predicted to kind of bend backwards into the amino acid binding domain, whereas in Pseudomonas it remains generally more on the axis of the native UDP manac substrate. Now, of course, this has also shown that the UDP has not reached the native UDP binding site, in fact, and instead is interacting with other residues, not other allosteric residues. So we haven't really been able to demonstrate a biodensity inhibition just yet. Now, the pattern of UDP binding sites for the other species has also been quite interesting. We got, especially for strep pneumoniae, where we got very little inhibition. There was notably the uridine domain, the uridine part is essentially, from the program suggests that it is just free floating. It has not given a suggested interaction with the strep pneumoniae. So potentially this might be a reason, might be an explanation as to why it's not exhibiting very much inhibition. But that's something to be, again, that's a preliminary prediction from this docking data. What we have got is having recently received the longer chain compounds with UP7 and UP8A, we have started getting some results which suggest that biodensity inhibition is possible. So structures which have been predicted to bind at both the uridine and the adenosine binding sites in Mercies specifically here. We're working on trying to get kinetic data on these to confirm whether this is actually, whether we observe that in the inhibitory modes, but yeah, that's something that we've been looking forward to if we are able to get the kinetics working on that front. As a kind of future perspective, we recognize that the dinucleotide phosphates are unlikely to be effective antimicrobials. They are, after all, found in the bacteria themselves. They have well-recognized synthesis and degradation routes. But what we are ultimately hoping for is to generate these, is to get an idea of the interactions and structural activity relationships that underlie this inhibition that we've observed, and which can hopefully be applied to compounds with other linkers which are more resistant to degradation, which are more likely to pass into the bacteria themselves as antimicrobials. So our aim is to look at potential hypothetical compounds which might mimic our polyphosphates as a scaffold, but that's something that we have not yet put, that's something to be done in the future. Yeah, with this powerful inhibition that we've observed, we've tried to look at getting crystal structures of these, which would be a very powerful result in checking whether our docking results are representative of what's happening. But we have not yet been able to get a crystal for Pseudomonas myrtae. We may, but for now, the current aim is to get a more comprehensive kinetic perspective of the sample space. So with other meta-enzymes, especially if we're getting more potent inhibition with longer chains, and also doing some stoichiometric work, because one thing we're not quite sure of yet is whether it is just this one molecule binding, or whether potentially you can have a molecule binding at the ATP sites, another one binding at the ATP sites, etc. That is the current state of where I'm at with the project. Yeah, I've worked a lot with Adrian and more on this. So, yeah.
Great. All right, thanks. I've got a very naive question. Sorry, so you're using the compound with 6 phosphates there, and the modeling studies have been done with compound with 3 phosphates. Sorry, so what is the assumption that it's just being cleaved there?
Oh wait, sorry. I should have made this clear. The predictive program I've been using was not powerful enough to model the entire 6 phosphate compounds at the same time. So keeping it in the same position, I split it into 2. So one with the one at the split into 2 at the phosphate chain. So to let it just model half of the molecule at a time, that it would to make it be able to accept the molecule. The entire hexaphosphate molecule was too big for it to.
I see.
Model.
Okay. All right, great. And I mean, obviously, as you lengthen the chain, you're developing a pretty polar molecule. Yeah. Is there any chance that the inhibition is because of some non-specific thing where the compound's just sticking to the protein? And do you have like a control compound where you've just got the phosphate chain with other things on the end?
We've had a look for that. It hasn't been too easy to source just a source just non-phosphate chains as a.
And they need to be synthesized, right?
Yeah.
Can I add something here? Stop sharing. In the initial work up to this, we tried AP4A, in other words, with adenosines at either end, as opposed to UP4A. And there was strong selectivity in favor of the uridine version of it, which kind of suggests that there is a specificity that goes beyond the fact you've got essentially a tetraphosphate there.
Yeah. Okay. Yeah. It's just as as you're increasing the number of phosphates, you got to kind of gradual improvement in the binding. And of course, it could be completely explained by exactly what you just said, but
Yeah.
could also be explained by just, you know, you're developing a snake-like extremely polar molecule that's just wrapping itself around your protein. I don't know.
Yeah.
Yeah, exactly.
So synthetically, Matt, if we could, we kept some charge to interact with the magnesiums, but changed the linker with something else to make it more cell permeable. Is there anything that immediately flashes to your mind or could flash to your minds corporately that we could even think of synthesizing?
Yeah, I mean, amines are going to make it pretty rigid and not very polar. The ethylene glycol suggestion you've got there is probably the one you'd immediately go for. So flexible and water soluble.
Mhm.
So the polyether chain, I would guess. Yeah, that would help.
Okay. All right. Peg.
Is that something that you could make, or is that something we're better off just paying for the CRO?
That's not trivial. The kind of the simple long chain without the phosphates is trivial, but as soon as you put the phosphates in there, it's more difficult. So it's good you've got a group that can handle those kinds of molecules, because that's not straightforward. We're having a real tricky time at the moment making a fluorinated ATP derivative for some NMR studies, and just handling these kinds of precursors is tough because they're so polar. So that, yeah, not trivial to make, but if your current group are not happy with making it, then we can always have a conversation about it, sure.
Okay.
Yeah.
Nice data though. Very interesting. Anyone else got any questions about that? All right, great. Thank you, Henry.
All right, thank you.
Warwick team, did you have any other updates you wanted to share about crystallography or evaluation of molecules or anything along those lines?
Well, crystallography, as mentioned before, I think I managed to get some things going. So I want to try the compounds without any dimeso and see what happens. The most potent compounds for the Pseudomonas myrci.
Right.
And then I also got, I saw Colette yesterday. I haven't seen her today, but I saw her yesterday, and she told me she has done the toxicity experiments for 3 of the AZ compounds, well, Yuhang compounds. I think it was 88, 86, and another 80. So we got data for that, and she was she had the data on the pen drive. So I am hoping to get it at the end of the day today. And then I can share with the rest of you. She didn't see anything like a strongly toxic, but we'll see once the data is analyzed. So it's not looking too bad.
All right.
She's also getting more tuberculosis strains or really mycobacterium strains, some of them more virulent and things like that. And she wants to try the compounds against those as well.
Okay.
Yeah. So we're getting ready on the mycobacterium side of things.
Okay. All right, great. And of course, as we've been corresponding about, the protein samples for the ASMS hit finding assay are basically ready to go. We're just trying to figure out how to get the data into the Toronto system, but those can be shipped and analyzed soon, which is fantastic. Yeah,
as soon as I think they replied already. So after the meeting, I'll have a look, and then I can continue with that.
Great, fantastic. All right, thanks. Guy, I don't want to put you on the spot, but did you have anything you wanted to share? We didn't talk about it.
No, not really yet. I'm still struggling with the chemistry. So yeah, so trying to get this done, which is basically the optimized series, aiming to improve the solubility of the compounds.
Okay.
So hopefully I'll have at least a few compounds next meeting, so I can actually present final slides. Yeah.
Great. No worries.
Fingers crossed.
It's been a nightmare.
Sorry about that. I think it's the LCMS issue.
Yeah, could be.
I know the pain of the Mur Ligases. You're not alone.
And anything anyone else wants to share? Because I think because we've been struggling with LCMS type issues this month, we haven't got a lot of new stuff coming from our side. I guess Yuhang, you did find a solution to the accumulation assay thing, right? Then you're about to send compounds for that in January.
Yeah, I'll reach out later because like currently our LC is still down a bit. Today, like I got like a flow cell blockage. I think it might just be ours. It might just be. I did analysis today. I think it might just be this batch of acetylnitrile. It's not quite great because like in industry, the side product acetylnitrile can self-aggregate. It can form polymer, so it's sort of like we constantly having blockage throughout the system, like part by part. So I have to take each part out, then deblock and check what's the cause what's causing that. So like now I'm suspecting that maybe it's just like this batch of acetylnitrile is having issues. We might need to do modifications on purchasing the solvent source or anything else, but after that we can sort everything out on the experimental side. So I'll reach out. I'll reach out to Crick. Yeah, actually our collaborators are Crick, and we were pretty happy to have them. I have a slide here. I think I got a couple questions, if you guys don't mind. Here you go. You guys see it?
Yep, see your screen.
Yes. I think I got two questions to ask because initially I would like to present our deck of biological data we have at the moment. For the alcohols, they were basically repetitions. The repetitions from the paper. There's only one novel that the paper doesn't have any data of it. But so
can you just zoom a little bit so that we?
Oh, sorry.
Yeah, that's it. That's it. Fine.
How about this? Yeah.
Yeah, it's fine.
So yeah. So these are the alcohol versions of the AZ series, and we are I'm not quite sure if we still need the data of the final two against E. coli efflux deficient. I think that's the TOLC mutant. The others were the wild type bacterial strains. Do we still need that data? I don't think. Not quite sure because I can't guarantee if the reviewer is gonna ask, why don't you guys do this as a comparison, you know. Also, like the essential parts we're missing is versus Pseudomonas, I think, because if we, I'm not quite sure if we still have the data
I think that
for.
was done before,
Oh.
but I will check with the girls.
Okay.
But I think it was done.
Okay. I can send the deck over, and I can print it out and send it over when.
Yeah,
I ship
if you can
compounds
send
to
it on
you.
an email, that's easier for us to share.
Yeah, absolutely. Also like when it comes to the aimings we made, I think there are two data I didn't have, which is this one, which I sent 2 years ago. I'm not quite sure if I missed that data or not, but yeah, this compound was set as a control to explore the solvent end, which were assumed as, and based on these data, do we still need the MICs versus Pseudomonas, or do you guys still have samples? Because I'm about to remake some of them this month or early next month in January. So if you guys need like solid samples, I can just add them in as a fresh one.
The only thing about that compound, right, is that that's the one that we originally designed to be to have the primary, the number 22. The compound, we originally designed that to be active and against wild type, right? Because that's what that aiming is doing. So for any compound, I mean, out of all of these, that would be the one where more information about would be useful.
And considering the fact, oh sorry.
Going back to WYH32,
32.
The missing compound, that is to say. So I've not come across it yet, but that's probably because it's probably changed hands a few
Yeah.
times. If you do have a fresh sample of it, then I'll
Yeah.
happily do the IC50 and the KI for you.
Okay.
So
Thanks.
if you add that to what you're going to be sending us,
Okay.
I'm sure that's done.
Okay. Thank you. Thank you so much. I know it's like it's not gonna be potent, but but yeah, it's we designed for that. We deliberately made it not potent. So
You never know.
yeah, I never know. Yeah, hope for the best. Yeah, who knows? One thing about sorry, I forgot to answer a question. One thing about these alcohols, because we can see the because our biological assays conditions are different from the paper. We can see there's a huge difference. There's a difference between the referenced IC50s and the experimental IC50s. Like based on the difference, can we make the assumption that the AZ595 amine derivatives is actually kind of potentially potent, It's like a 10 times or 100 times. If we divide by 10 times, 100 times, it actually was having something. Okay.
So the reference that this basically pertains to the Mercy paper. Some of those IC50s were actually below half the enzyme concentration, which makes me wonder about how accurate some of them actually were. All I can tell you is that the data we've given you is internally consistent.
Yeah.
But to precisely try and marry one set with another, apart from general trends, I think is difficult. I mean, at a case in point, IC50, Pseudomonas aeruginosa, WYH78, less than half a nanomolar. In actual fact, the concentrations in that paper were single digit or more nanomolars with regard to enzyme. So, like I say, it's difficult to be sure about exactly what's going on.
Okay.
So all I can tell you, like I say, our data I'm perfectly happy with.
Yeah.
I think it is internally consistent, but trying to make comparisons with the AZ paper, I think is tricky.
Okay. I'll just ignore that then, because I was about to add it to my chapter, but thanks for the question. But thanks for the answer. I probably shouldn't. This is not a scientific thing.
Yeah.
Thank you. And here's I'm about to make this guanidinium derivative of the AZ595, and hopefully I can ship it soon. Also this 87, because 88 is having a weird potency against this microbacteria or anything. I think it would be good to make the initial pair. So I just decided to make it, and hopefully this week as well. I'm in progress of remaking some 88 just in case you guys need more. That's also some questions about this compound, whether we need more of it, whether we need the MIC data of this compounds. I somehow didn't have the data. I'm not sure. Maybe it's just my problem, but I would like to double confirm if it really hasn't been tested.
We will check.
Yeah. Thank you.
Was it surprising? That for me is the surprising one. Right?
I think it's been done, but so many numbers.
But it's a big change to the structure, right? Massive change, and it's fairly rigid, and it's surprising that it's surprising it's so effective.
Yeah. Also, finally, we have these we have these structures, but I'm not quite sure if they are worth to do the MICs because especially yeah, they're not essentially that bad.
TR6 and TR64 and 68, those
Yeah.
have they been sent already?
Yeah, have been
Okay,
sent
I
already.
think
Yeah.
they might be already tested. Yeah.
Oh, okay. What about 16
think-
byproduct 70?
I think as well. But I'll
Oh,
double
thank you.
check again with them.
Yeah,
Once
yeah.
you send me this, I will check with the girls.
Okay, thank you so much. Thank you so much.
Okay.
I'll drop the link. One final question. Was Matt asked me previously to ask you guys. So we have these AZ compound being shifted from AstraZeneca for crystallography or some or some inhibition versus other Mur ligases, I think. But just asking, by any chance we have them as we have the IC50 data versus Pseudomonas VOC, because we would like to see the if their data also matches with the original paper, but just in case. But it's okay, like if you guys don't have it, because yeah.
We got some data on them, right?
Oh, okay.
Yeah, I'm trying to
But I can't remember what exactly.
Oh, okay. Okay. I'll
Were these the ones that were shipped from the company?
Yes.
Yeah.
Yeah, so yeah, I mean, obviously for inclusion in the paper, it would be fantastic to include those because we got samples for free.
Yeah. This was from Anita.
Yeah, that'd be it. Okay, we'll chase that down.
Yeah, we
Great,
chased
thanks.
this down.
Yeah.
Thanks a lot. Fantastic. All right, thanks,
Thank
Yuhang.
you. Thank you so much. Yeah, that's all.
Great.
Yeah.
All right. Any other business? Christy, do you want to add anything?
Yes, that's okay. Just kind of overview. I had a really good visit to Mark Sutton and other members of UKHSA in Porton Down, talking to them about open innovation, because they have an open innovation platform for biology. They like very much what we're doing in open antibiotics. They also like what we're trying to do with something called the LEAD program, which is Leadership in Enhancing Antimicrobial Discovery, which is part of what we're doing with UK and Japan. I'm going to Hyderabad and Bangalore next week to talk to the bioinnovation sector there across policy and industry and academia around that, because things are progressing quite fast in India at the moment around antimicrobial discovery and development. So you have Wockart and Bugworks and others coming up with new molecules and partnering with GARDP and Carbex. And around Hyderabad, they're really pushing that region, like Cambridge, as the bioinnovation hub for India. So I'm just going to talk to them about what we have in the way of this kind of proto-consortium of open innovation and discovery. So that's just an FYI. In January, UKHSA are so engaged now, they're wanting to pull together a meeting of all the public sector actors all the way through to MHRA to try and defragment antimicrobial activity and pull that all together in a more cohesive manner. So the aim would be to get all of those public sector grouping together with government, with Department of Health, with Department of Trade, Industry, Science, Innovation side as well, just thinking about how we think about a new model for discovery. So that's going to happen. So my question, Matt, is do you do anything with people in Hyderabad and Bangalore at all? Do you have any connectivity there already?
No, we spoke to Bugworks many years ago about doing something, and there was certainly interest in having one of their series hosted in an open project, but it didn't progress anywhere. And I think that was partly because they were so busy trying to raise money, that they had bigger things on their mind. Hyderabad, no. We have lots of links over the years with the CSIR group in Lucknow, because they hosted a lot of open stuff maybe 20 years ago or so. When we were just starting the malaria project, they were doing a TB project. But nothing, we visited and we had a great time, but nothing formal came out of it. And it was always the intention to make links with those groups, because I sensed that underneath the surface there was a lot of interest in the people in the labs. Sorry, a lot of interest from the people in the labs in working in a more open way. In some cases, there was still IP skepticism, as you might expect.
Great. So we have connections with that group in Lucknow as well. We can't physically get there this time, but we've got roots there. Ditto, Bugworks, Dr. Reddy's, Wockart and others. So I think it's interesting. So I just want to touch base with you now about that.
Great.
So I'm just trying to think in the UK now that UKHSA are involved. Who else would be, if we thought of a virtual hub within the UK for the discovery sector, beyond the chemistry that you do, what we do, what Portland does, which is quite modest. Their open innovation platform is no bigger than the AMR screening facility that we have. It's basically 2 full-time funded people.
Yeah.
But it's government, and I think that's really important to persuade government to do something more, rather than untrustworthy academics who are only self-interested, apparently. You need a state actor to speak. So I think that's where their influence will be. I was going to have another chat to Annette von Delft down at Centre for Medicine Discovery at Oxford, because they're trying to get the platform, their discovery platform, which is mainly viral at the moment. But I could see them working obviously closely in various ways with synchrotron by Frank.
Indeed.
Just somehow trying to kind of get this kind of web of activity between synchrotron out through chemistry, biochemistry, antimicrobial testing. So if you've got any further thoughts on individuals, groups and things who are like-minded, who are like an open way of working, then just do message me.
Yeah, we'll do. I mean, yes, absolutely. So Annetta and Frank's team, we're in touch with a lot because of the viral work. And so there's a lot of interest there in joining forces to build something bigger. The guy that we're working with on the accumulation assay in the crick, that assay is pretty high throughput and interesting. We're about to send our first compounds, but as a resource, that is something I have not seen anywhere else. So there could be an extra thing there that's based at the crick that could be of interest.
It would be good, because LifeArc and crick are almost welded at the hip. And LifeArc will be a key funder side in this. I mean, their current chief executive is from the crick. They've got other senior managers from the crick. They're going to relocate next to the crick.
Yeah, I heard that. Yeah. I heard it. I mean, the two other things I was going to mention. One was, I mean, we're working with the UK HSA on this 96 well plate project. We sent them 96 compounds just from various projects, and they've been screening them, and we're trying to show that this kind of activity is quite easy to do in terms of putting data in the public domain on bioactivity of compounds that would otherwise sit in fridges. So they've screened our molecules against a few different things, and we're going to talk about the data when they are done, possibly in January.
Is that through that iFast platform?
No, we just sent them. We just sent the compounds and said, do you fancy testing these? And they've been working on them since then. So it's not through anything formal, other than the fact that we have good research links with Joanna Bacon.
I think if it's a 96 well format, they co-invented iFast with I think they'll be plugging it in just when you said 96 well. That's
okay, great. Well, yeah, no, I mean, they seem to be moving pretty fast on it, so it's good. And the other thing I was going to mention was the thing that we're sending the purified protein samples for this affinity selection spec screen. So if on your travels you come across any groups there who also are doing a very target-based approach and have a new protein and want to get it screened, then there should still be capacity for doing that. It's a perpetual problem, getting a screen done for a decent amount of money, and finding molecules that are soluble and commercially available. So yeah, anyone who is interested is please put us in touch. One of the reasons I mention that is because the big focus of the SGC is on human proteins and human disease. But there is, I mean, essentially the technology is the same. So there is a huge amount of interest in microbial targets, not least because you can imagine all kinds of things that you could do to understand or probe microbial communities that might be of relevance to climate change. So anything in that area is of interest.
Remind me, Max, I've already forgotten. Where is the proteomics facility based? That's going
So
to
this is a screen that's done that is mass spec based, which is done in Toronto.
Toronto, okay.
Yeah, there is a fancier thing that relies on a DNA encoded library screen, which has a machine learning element, and that is mostly happening in Chapel Hill. And if the SGC plan goes well, then we'll be able to do all this stuff at UCL.
Cool. Very good.
But you need protein sample, and it needs to ideally be a new protein, right? Where there's not much hit chemical matter, or in an ideal world, a protein where the function of the protein isn't known. So it's trying to understand biology better. That bumps things up the list.
Okay. Cool. Very good.
Great. Well, safe travels. Sounds really interesting.
Thanks. Safe microbiology is what I'm hoping for.
All right. Nice to see everybody. I guess we will surely have a meeting in the new year, but we can fix that up depending on people's how people are dealing with the post-Christmas effect. So up until then, have a lovely holiday and be in touch with you all soon.
Yeah, friend.
See you soon.
Bye.
See you soon. Bye.
Bye.
Bye.